We're excited here because we are a clinical research center that's about to start up our COVID-19 vaccine program. And so we're trying to reach out to community members and help educate them about what's going on with the science specifically related to the vaccine. So I'm going to give a little background in terms of where we are with the COVID-19 and vaccine development, and it's an informal session, so please feel free to stop me and we'll definitely have some time for discussion moving forward. So first, most people have an understanding what a virus is, but it's a good idea to just review it. So the best description of a virus that I've seen is from the biologist Peter Medawar, who said that a virus is simply a piece of bad news wrapped up in a protein. And it's a very apropos description because viruses are basic particles that are barely alive. In fact, they don't even have enough genetic information to run their own business. In order for them to run their day-to-day business of surviving, they need to capture the mechanism of another cell. So viruses actually can't live by themselves. They can only live within other cells. So you put a raw virus on a surface and it won't survive for more than seconds without the functions of other cells to help them carry on their basic needs. But they are very effective at getting into other cells, including human cells, and that's where they cause trouble. So this description is really very apropos, and we'll talk more about certain viruses that either can or cannot infect human beings, why this understanding is important, and in particular, why this understanding will help us develop a vaccine candidate moving forward. So we're all in this period of time where we have a surge of COVID-19 in our community, unfortunately. There's good and bad news. The good news is that it's moved from an older population. 3 months ago, the median age for patients with COVID-19 in Florida was about 60, and now that's down to about 30. And the reason it's good is because this virus has particular characteristics that seem to favor people who are younger. So the mortality rate for people who are 30 and younger is actually less than 0.1% in the state of Florida, which is really fortunate. Above 60, it's a different story. Then we're talking about a 3% or so mortality rate. And if you go to New York and some of the places that got hit first, they were looking at mortalities of 10% or higher. So there's something intrinsic about this virus that seems to have older people do worse. Now, not all viruses are like that. So just as a historical reference, the worst flu epidemic to hit the United States, and really the worst viral outbreak in the United States, was the Spanish flu in 1918. The Spanish flu had such a big impact that it was the only year in US history where the population of the United States went down year over year. There's never been a year before where that happened, except for 1918. And if you go to cemeteries that are 100 years old or so, you notice that the people in the cemeteries are all young. So that particular virus hit younger people much worse than older people. So people in the military, for example, between 18 and 24 had a 7% mortality rate. There's a fort in Massachusetts called Fort Devers, and they created this field hospital, and they had 14,000 young men between age 18 and 24 in that hospital with influenza, and 750 of them died. So that's 7% mortality. It's pretty incredible when you think about it. This virus is different. This virus, COVID-19, has that effect on older people, not younger people. So the fact that it's moved to a younger demographic is a blessing in disguise in that at least those people don't get as sick. And to give you some insight, I worked at Memorial Hospital this past weekend, and we've seen a big increase in the number of admissions with COVID-19. But we've had patients there all along. So 2 or 3 months ago, we had just a handful of patients, maybe 5 or 8 patients, but most of them were on ventilators. Over this weekend, we had 50 patients at Memorial Hospital with COVID-19, but only 1 was on a ventilator. So we've learned a lot over the last 3 months, including how to treat these people. When we were first dealing with this illness, the thinking based on some European information was that putting people on ventilators was a really good idea as quickly as possible. We've now learned that it actually makes things worse. It actually makes the inflammatory condition exacerbated, and people that get on ventilators often don't get off. They succumb to the illness before they get off the ventilator. So now we've completely changed the tactics, and we're putting people on high doses of oxygen without putting them on ventilators. It's only the last resort. The other thing that's changed is that we have drugs now that we know are effective. So there's a drug called remdesivir. Have you heard of that? Yeah. So remdesivir was actually developed for Ebola virus, which has a similar characteristic to COVID, but it's much more lethal. Ebola untreated has a 90% mortality rate. And remdesivir was developed for Ebola and shown to have some benefit for that. So that was actually on the shelf when COVID-19 hit. And we actually did clinical trials with it to show that remdesivir works. So that's very different and very apropos to what we do here. So when you first were hearing stories about COVID-19, you're hearing about hydroxychloroquine and zinc and other things. And none of those things were studied scientifically. Doctors were just trying them. So the hydroxychloroquine story started because some French physician tried it on a bunch of patients, and he thought that they had better outcomes, but it wasn't a controlled clinical trial. And I and a lot of other people were very skeptical because hydroxychloroquine has some anti-inflammatory properties, but it's basically a drug that we use for lupus and gout and things like that, not for infectious diseases. It doesn't have any anti-infectious disease properties. So we were skeptical. And sure enough, when they did controlled trials, there was no benefit to the drug. So when people were treated 2 or 3 months ago, we were putting them on hydroxychloroquine and Zithromax, which is an antibiotic, but not an antiviral. And those didn't particularly work. Now, when people come in, we're putting them on remdesivir, which is a specific antiviral and prevents the virus from replicating. And they get put on high doses of steroids that have been shown to have a big impact on the inflammatory system and reduce inflammation. And that works. These are now proven therapies. So the outcome of patients once they come to the hospital has changed quite a bit. And if you actually look at the mortality statistics for Florida compared to New York or Massachusetts, you'll see that the mortality rate is much, much lower in Florida than it is elsewhere, in part because we had time to learn how to treat patients better. And of course, also the fact that it's in a younger population in the second wave. The other thing that's interesting that you don't read that much about in the news is that we're already in the second wave of this illness. And what happened is that COVID-19 started in China and then came into Italy because there's a lot of commerce between Wuhan province in China and Italy. In fact, all the Italian leather makers and other people go to China to get some raw materials and things, and they put the finishing touches in Italy. So there were literally, dozens of flights a day between China and northern Italy in the industrial zone of Italy. So that's the first place that got hit, and then from Italy, it seeded the rest of Europe. And the first Americans in the Northeast were mostly seeded through Europe, and that turns out to be one strain. The other strain came from China through the West Coast, mostly to Chicago, because of the international flights between China and Chicago. And that strain now has taken over. And 90% of the cases in Florida and many other places in the United States are the Chicago strain, not the New York strain. So there are some subtle differences here that you don't hear that much about. There's a debate about whether or not one strain is worse than the other. The differences in the genetics of the two strains is relatively subtle, but for whatever reason, we're seeing less mortality, less death rates here in Florida and other places that have been hit later. So part of it may be strain differences, part of it are probably demographics, and part of it is just that we treat people better. Some combination of all those things. But we know that it's much better to prevent a disease than to have to treat it. And so we're excited about the fact that we'll be starting vaccine studies in about a week. So we're really, really excited about it. And we've been given a grant, we've been awarded a grant to treat up to 500 patients here in Jacksonville with the vaccine. And the vaccine that we're using uses a completely different technology than previous vaccines, which is really, really exciting. And we're working with 2 different companies. Both of the companies have released their phase 1 data. So we now have information in about 200 patients. And it turns out that so far, so good. So far, these vaccines seem to confer immunity at least the same level as somebody who's actually infected, and maybe even more. But the proof is in doing clinical trials, being scientific about it, and seeing what happens when you do a structured experiment. Otherwise, it's just guesswork. So we're really excited to be part of that. And we'll get to that in a second. But I want to give you a little bit more background so you understand why this technology is kind of a breakthrough and the differences in different vaccines. So the way vaccines are traditionally made is that you have to grow a virus in some sort of medium because viruses can't live on their own, as I mentioned. So the most common way of doing that in the past has been to grow the virus in egg cells. And there's, it's worked for a lot of viruses, including polio, et cetera. But there are limitations. One, the virus tends to grow relatively slow in egg cells. And two, it's a difficult task to isolate the part of the virus that creates immunity from the other parts of the vaccine preparation, including the egg proteins that some people are allergic to. But basically, the first step in vaccine production is to grow the virus. By the way, we've been very involved in vaccine studies in the past, and one of the things that we've done that was considered a breakthrough before this latest technology came to be is that you can actually grow the viruses in things other than eggs. So tobacco leaves is another way you can grow viruses. We actually did a study about 5 years ago showing that the flu vaccine can be grown based on viral cultures in tobacco leaves, which created fewer allergies and actually a faster and a more efficient industrial process for creating the vaccines. So there are other media that can be used to grow the virus. So once you grow the virus, then what you want to do is you want to isolate the antigen, which is a medical word for protein, which is what the body identifies and creates immunity against. So that antigen can be the entire virus, which is almost never done, or a piece of the virus, or most commonly a virus that's been inactivated in some way, so it can't reproduce in human bodies. And once you give that antigen, then your body is aware of what it's trying to fight, because the body is able to identify human proteins from non-human proteins, and the immune system gets activated when it sees a non-human protein. Once you identify the antigen, then you have to purify that antigen. So what that means is that you don't want to give a lot of contaminants to people. You want to just give them what is going to trigger the immune response specifically against the pathogen that you're interested in. So that becomes a very complicated process of just separating a pure product, which in this case is an inactivated virus in that cartoon. And that is a very, very important step in terms of creating something that can be used in patients. Finally, you want to make sure that the virus is dead. Obviously, the worst thing that could happen is that you pick out the virus and you think you inactivated it, but it still has some activity and it can infect human cells. That would be horrible. So a lot of effort goes into make sure that the virus is completely inactive. Sometimes radiation is used. There are all different kinds of methods to make sure the virus can't harm normal cells. And then finally, once you have the purified antigen, then you need to put it into a solution that will ultimately be injected. And that includes using stabilizers so that it doesn't spoil or doesn't get infected with other bacteria or other organisms. So there's a whole very complicated process to make sure it's completely sterile and something that's going to be as safe as possible for human beings, including testing for trace elements of some contaminants. And then once you get your manufactured product, then you got to get it to the consumer. And that in of itself is a big task. You have to go from a huge production vessel to vials, to trucks, to a doctor's office without creating any contamination or spoiling of the product. So that's kind of how vaccines have been traditionally made. But there has been actually a breakthrough over the last 10 years based on a better understanding of genetics. And we're really super excited to be part of this process. So what we've learned is you can actually tell the body how to make a protein. So if you give the body a genetic instruction just to make a specific protein, your body will do that. And we actually have drugs now that are utilizing that concept, that you can use genetic information or genetic code in order to get the body to do something. So this new technology is not giving you a virus. It's not giving you part of a virus. It's not doing anything but giving a set of instructions for a body. And in the case of COVID-19, that set of instructions is saying, okay, we're going to produce the key critical piece of the spike protein of COVID-19 that allows this coronavirus to infect human cells. And the reason we're doing this is that the body is smart enough to know that that is a protein, that is a viral protein, a foreign protein, and it will create an antibody to prevent that protein from infecting the cell once it's identified by the body. Okay, so it's the coolest thing. Basically, these products that we have are very small pieces of genetic material in a little bit of a fat particle. They're injected. The body will make the protein. You're not getting the virus. You're not getting anything that could possibly be infective. And the body creates that and then immediately creates immune response to it. So just absolutely crazy breakthrough technology. Yes, sir. Yes, exactly. Yeah, the coronaviruses are characterized by these little spike proteins. So remember, there's a lot of different types of coronaviruses. This is a common misperception. So when we say flu virus, that's multiple viruses. Influenza virus is a whole family of viruses. And then we have the H1N1 flu or the Spanish flu or the swine flu. These are all types of influenza virus. Coronaviruses are ubiquitous. They're all over the place. 99% of them cannot infect human beings. But when there is a mutation of this protein that allows it to interact with a human cell, then it can start to infect humans. And for COVID-19, that spike protein is able to interact with a receptor in the human lungs and in our nasal passages called the ACE2 receptor. And that's what it allows it to get into the human body and into human cells. So the concept is to block the virus's ability to infect human cells using antibodies. Yes. Moderna, yes. We are working with Moderna and Pfizer. They're two different consortia. I'll get into a little bit more of that in a second, but yes, we are working with Moderna and with Pfizer. Yes. The immune response? The immune response, I'll show you more of that in a second, but basically, there are molecules in cells in our body that are responsible for fighting infections. So we have intrinsic ways, we have ways that we're born with that help us fight infections. And what the vaccine does is it activates those pathways in human beings. And I'll explain it a little bit more. Hopefully it'll be clear by the end. So this is a refresher of high school biology. I think it's probably now taught in grammar school. It's probably taught in college when I was going to school, and then moved to high school. And now it probably is like third grade biology. But if you remember, you have DNA, and DNA is the genetic code for all cells. And then DNA codes for certain genes, and the genes then code for proteins. And they code for proteins through something called RNA. DNA has 2 strands. RNA has 1 strand. So both flu virus and coronaviruses are RNA viruses. It means they only have 1 strand. There are some viruses that are DNA viruses with 2 strands, but these are 1. So once you make an RNA strand, that RNA can go to the ribosome, which is a part of the cell, and the ribosome reads the code and then produces a protein. So what our vaccine does is it goes to the ribosome and it creates a spike protein that the body can identify. So it's primed, so it knows who the bad guy is. So that's just a little refresher of how our bodies work. Does that make sense? All right. So there are currently, as we speak, so first of all, there has never been in the history of medicine so much firepower put onto a problem. It's unbelievable. There's incredible amount of resources being spent and so many brilliant people around the world who are working to fight this thing. And so I'm very confident that we will have a functioning vaccine within a year just because there's so many people working on it. So something is going to work. So keep in mind, there are actually 145 COVID-19 vaccines already produced. Can you believe that? It's crazy. Of those, about 16 have already been tested in humans. Okay? So the 16 that's been tested in humans, the 3 that are ahead of the pack are the vaccine that was developed at the University of Oxford, and that one involves what's called an adenovirus platform. And an adenovirus is a common cold virus, and the researchers at Oxford inactivated the adenovirus, and they tricked the adenovirus into producing the spike protein. So they basically put the spike protein in the adenovirus, and they created a vaccine based on that. So that's actually an injection of a virus that's been inactivated. So there's pluses and minuses of actually injecting an entire virus, but that's that platform. And they started their human tests probably in late April, early May, and they just started their phase 3 studies at the end of June, and they've been doing it for about 3 weeks now. They already have 8,000 people dosed with the vaccine in Great Britain. So that's the furthest ahead. Then the 2 that we're working on here in the U.S., that's not in the U.S., by the way. That's not available in the U.S. The 2 in the U.S. that are most ahead are the Moderna vaccine and the Pfizer vaccine. And they both use this messenger RNA platform. And as I mentioned, the phase 1 studies have been completed for both of those. And so there's been about 200 Americans dosed with those vaccines, and we'll be part of the phase 2 and phase 3 trials. And the goal is to vaccinate 30,000 Americans in the next 3 months. And so we, here in Jacksonville, will be a big part of it. We're scheduled to get 500 patients involved. So that'll be 1/60th of the entire program, maybe even a little bit more. And so we're one of only 100 centers in the country that have been awarded this program. So we're very pleased about that. And a lot of those centers were NIH centers, federal government-sponsored centers. So we're probably one of about 30 or 40 private centers that have been awarded the vaccine study. So we're pretty proud of that. But again, we don't have any vested interest in the results per se, other than just being good citizens and wanting to see this whole problem go away and have a solution for it. But our job is to have a very safe experience, as safe as possible experience for our patient population and for our community, and most importantly, to report the information to the Food and Drug Administration. So that's what we're here for, and that's what we got a study grant to do. So just to be clear on where we are in terms of vaccine development and our role in it. That all make sense? Okay. So as I mentioned, the phase 1 results of the RNA platform vaccines have made the news, and people are pretty excited about that, but there's still a lot of work to be done. And as I mentioned, there's still a lot of people working on this problem, and we don't know which one's going to be the best vaccine. But we're really excited about the mRNA concept because you don't actually give a virus. So just to give you a little perspective, typically it takes about 5 years to develop a vaccine. And so if you go back to the avian bird flu from 1996, they didn't have a vaccine for that until 2007. So it took a long time to develop that. Now, on the other hand, again, this is H5N1 flu. On the other hand, once they developed that, it took a shorter period of time to deal with H1N1 flu, or the swine flu from 2009, because they had already developed the vaccine. They just needed to tweak it a little bit in order for it to be commercially available. So once you have something on the shelf, it makes a big difference. But if you don't have something on the shelf, it typically takes a long time. SARS is a corona-type virus, and people are working on a vaccine, but fortunately, this is a much more lethal disease than COVID-19, but fortunately, it kind of died down before a vaccine was developed. Same thing for MERS. You can see Ebola, which is completely devastating. There's a 90% mortality rate for Ebola, and that took over 5 years to develop a vaccine for. So here we are in COVID-19, and literally within about 5 or 6 months of discovering the virus, we already have 16 vaccine candidates in clinical trials. So there's never been anything like this in the history of medicine. So again, it's exciting and a testimony to the fact that we've never put so many resources into solving the problem from the medical standpoint. This was an article from the New England Journal that showed the fastest vaccine prior to COVID-19 was 5 years, and now we're talking about doing it in 18 months. And probably, my guess is we'll have it in between 12 and 15 months, something commercially available. But keep in mind that even though we have a vaccine, we don't know how long that immunity lasts. So if we give a vaccine and you're immune only for 2 or 3 months, that's not a very good vaccine. We need a vaccine where we know that the effect will last for years. No, again, that was only launched about a month ago. So it has like about a 6-week head start over the mRNA, but it's not that much ahead. From the time that the genetics of COVID-19 were identified, which was probably about March. So how do you become immune to a virus? Well, there's really only 2 ways. You either get the disease and you recover, and then your immune system knows that that's something that doesn't belong there, and your immune system develops a memory and will protect you from that, usually for the rest of your life. Or you can get a vaccine, and that will elicit the antibodies that will identify and kill the virus, and that will in turn have the immune cells that fight viruses, become activated if you get infected or if you're exposed to infection. So measles or polio, there's a lot of things are good examples. Once you get vaccinated, you're protected usually for life, unless your immune system just completely fails. So you've all heard about antibodies, and they can be very confusing for people. So this slide's help you look at that. So if you get infected with COVID-19 on day 0, your body starts its immune response usually about 5 to 7 days later. And the first thing that it does is create something called IgM, which is a very large molecule, which is the body's first effort to identify the exact problem that is causing illness, and then to create these proteins that offset that particular protein, that particular viral protein. And then IgM tells the body to produce something called IgG, which is a much smaller molecule. This is about 5 times bigger than this molecule. And the IgG is smaller, it's more mobile, and it can be stored in your cells as memory. And the ultimate concentration of IgG is much, much higher than IgM. But that only starts about 2 weeks after you get infected. But hopefully you have a good response here, and hopefully that response lasts for years. And even years and years and years down the road, you hope that you have enough IgG in memory such that if you do get infected, instead of having to wait 14 days, almost immediately the IgG kicks in and prevents any serious illness. So antibodies look like this. They have a part of them that's just a structure that's similar for everything. And they have a part of them that is very specific for the protein that you're trying to neutralize. So ultimately, what you're trying to do is you're trying to prevent the green guys, the green spheres, are the bad guys. And the antibodies are interacting with the green spheres so that they can't affect the cells. We have another picture that I think is a little bit better than this one. So there's 2 types of antibodies. One's called a binding antibody, and they're often produced at high levels, but they may or may not produce immunity. And the reason for that is because they don't hit the virus in the spot that's critical for its interaction with human cells. They may hit the virus someplace else. Then you have neutralizing antibodies, and these have the capability of neutralizing the virus. So you probably have heard about hepatitis B and hepatitis C and HIV. And they're all viruses, but there have been different approaches to the treatment. So for hepatitis B, this is rarely a concern anymore because of an effective vaccine. So all healthcare workers like myself and most people in the community get hepatitis B vaccine. And once you get hepatitis B vaccine, you're protected against the hepatitis B virus. Hepatitis C, interestingly, is another type of viral infection that's pretty much been eradicated, but that was eradicated by drugs, not vaccines. There's no vaccine for hepatitis C. And the reason for that is because hepatitis C doesn't come outside of the cells. So most viruses, when they infect a cell, they eventually kill the cell, and that releases the virus, and then the virus goes to other cells. But in hepatitis C, the infected cell merges with another cell, and the viral particles can go from cell to cell without going into the circulation. And it does that in the liver, of course, hepatitis being a liver infection. So in order for vaccine and antibodies to work, the virus has to get into the circulation, because that's where the immune cells are doing most of their work. HIV, we don't have a vaccine for that, but we have drugs that have basically neutralized that problem. And for HIV, the tricky issue is that the virus actually attacks the immune cells. So it attacks what's called T lymphocytes and makes the T lymphocytes incapable of doing their job of activating the immune system. So the reason I'm pointing this out is because there are uncertainties. We think a vaccine is going to work, but there have been other viral illnesses that we have had to treat with drugs rather than vaccines. But from everything we know about COVID-19, it seems to be more like the flu than like HIV or hepatitis C that should respond to a vaccine approach. But that's one of the things that we're learning as we do these clinical trials. The other thing is that, as I mentioned, coronaviruses are everywhere. And coronaviruses are most commonly found in pigs and bats and rats and many organisms. And they usually cannot infect humans. But every once in a while, they have a mutation that allows them to infect humans. And that's what we're dealing with in COVID-19, something that was in an animal community that change and then infected humans. So I mentioned about sort of a visual so you can understand what neutralizing antibodies can do. And by the way, both the Moderna and the Pfizer platforms have proven neutralizing antibodies. We've already had that information shown to us. So a binding antibody, this little blue thing is an antibody. The green little house is a virus, and this is a cell. So if the antibody combines the virus in a place that's not the location for the interaction between the virus and the receptor, it may get in the way a little bit, but it's not going to block it completely. Whereas if the antibody hits the virus in a place where it has to combine with the rest of the cell receptor, then it neutralizes it and makes the virus unable to infect further cells. Does that make sense? Okay. So this is what we know. We know patients who recover from COVID-19 usually mount extremely high titers of neutralizing antibodies. We know that people differ significantly in the amount of antibodies that they make. What we don't know is the duration of this effect. Will it last for a month? Will it last for a year? Will it last for 5 or 10 years? Obviously, we won't know if it's going to last for 5 or 10 years anytime soon, but we should hopefully know if it's going to last for a year based on the studies that we're doing over the course of the next several months. As I mentioned, never in the history of medicine has there been this level of intellectual firepower focused on a disease. So that's exciting. So again, as I mentioned, we're doing these educational sessions for the community. One, to get people aware of what the issues are, to talk real common sense with people. Unfortunately, a lot of stuff that you get in the media is a bit sensationalized or colored by political opinions and other things. And we're here just to make us, to be part of the scientific advance. And so what I like to tell people is that you're not guaranteed to get the vaccine in these programs. We can't guarantee that they're going to work, but we will tell you that you will become ambassadors of knowledge. We all will be that. And we will all be contributing to this effort to understand how to fight this scourge. And that we can certainly speak highly of and tell you that we'll do everything possible to make this experience a good one for you and also help you learn about things, avoid COVID-19, hopefully. we will talk about hygienic measures that are shown to be effective and also hopefully get people involved in the vaccine program where a lot of you will get the vaccine, but not everybody, and we'll be monitoring you very, very carefully. Yeah, any ethical, properly scientifically conducted clinical trial will have a placebo group, and these studies will have placebo groups. Yeah, so we've had a really good response, and as I mentioned, it looks like we will have the ability to dose 500 people in Jacksonville, and we probably had about 400 people that have already kind of called and volunteered, and maybe not all of them will end up doing it. Plus, we wouldn't recommend it for you until we looked at your medical history and know a little bit more. If we look at your medical history and we see something that makes us uncomfortable with you participating, we're going to tell you that. And sometimes people get disappointed, but it's we're kind of looking out for our community. But there's not that many restrictions for the program. So if you're on steroids, for example, you wouldn't be a good candidate. But if you're somebody who was at some risk, either through occupational exposure or because of age or because of concern that you interact with people who may not be taking the proper precautions, et cetera, this might be a really good program for you. And one of the nice things about the program is that there will be measures in place to monitor you. If you feel sick, you can come in, we'll check you out, make sure you don't have the virus, that sort of thing. Well, the study is anticipated to be at least a year, and it could be longer than that, but it depends on what happens in the world. So with all these studies, you can imagine if Moderna looks at their results in 6 months and they see an overwhelming effect of their vaccine, well, they're gonna have to tell the world about this, So that's how these things work. There are ethical principles involved, but the study could be at least a year and it could be longer. If let's say the vaccine is just a little bit effective, but it takes time to show that, then it may be more than a year. You get at least 2 injections. Yeah, it depends on which program you're involved. In the beginning, we'll see about weekly, but every program is a little bit different. And some of the visits are just telephone visits. We'll call you, make sure you're doing okay. And some of the visits will be in-person visits. But if you guys are interested, then we'll sit down with you and give you a consent form and go over all the details of the program and lay out exactly what the expectations are. But with all the studies, the feeling is that you need 2 doses to become immune, and that you will definitely be monitored on a regular basis, and it'll be more frequent right after the vaccination, but then it'll be mostly just keeping in touch to seeing how well you're doing. Well, again, we don't want, not everybody's going to get the vaccine, so we don't want to put you in harm's way, but we happen to know that just by living in Florida, people will be exposed. So we don't want people to go out and seek the infection, or to you say, oh, I'm in the study, you can't hurt me. We are not advocating that whatsoever. We're advocating being smart, social distancing, taking proper precautions, proper hygiene, and hopefully keeping our patients out of harm's way. Having said that, we also know that some people will get infected, and that we're hoping that the people who have the vaccine are less likely to have significant illness. We will be blinded to that, but the blood test will be done. Yes, that's part of it, and part of it is to look for different antibodies, and this gets very complicated and sophisticated, but there are different antibodies, and the difference between a binding antibody and a neutralizing antibody is not something that you could get at Quest or any local lab. That's done at a very specialized research labs. So if you go to Quest and you get an antibody test, you won't know if it's from the vaccine, you won't know it's from exposure in the community, or you won't know if you just intrinsically have been previously infected with something that has cross-reactivity with that with the COVID-19. You'll be given that as part of the program, yes, but we won't know the results. Yeah, obviously you would have the advantage of knowing if it works or not. If you're at 13 months and it doesn't work, then we wouldn't make that. We wouldn't offer that to you. And part of this is just based on anticipation of what we're going to find. But I've never been in a study before where something that worked was then not made available to the patients who participated in the trial. Again, that's not a guarantee because we don't know the details. But based on experience, if something works, then all the participants are allowed to then get the active vaccine. But that won't be a quote guarantee anyplace. We're allowed to put people in between 18 and 85. I think everybody in this room is in that age group. Yeah, so again, we already have about 200 patients worth of experience, and I get briefed on this on a regular basis from the companies, and there has been no serious adverse events. That means nobody's gone to the hospital, no one's gotten super sick, no one's died. So there's been no real serious complications. There have been some skin reactions, and that's one of the things that we talk about is whether or not that could be a tip-off in terms of whether or not you got the real thing or not. I won't get too much into that, but there'll be a control group that will probably be less likely to get those sorts of things, but that's part of doing research. But having said that, there have been no major issues, but there have definitely been some minor issues. If you guys are interested, we'll give you an appointment for within the month. We'll probably be dosing our first. I'm going to get the final authorization on Friday, and then right after that, we'll be dosing people. So I think by next week. Again, we have to know your medical history. So some of you may already have your history in our system, but for those that don't, please fill out the forms, and we'll go over that with you. So just make an appointment. If everything looks good, you can sit down with one of our folks here, and if everything looks good, set up an appointment to get dosed. And again, the first date of dosing is still not 100% certain because I get the final briefing this Friday, and then usually you can start within a day or two after that. So probably we'll be dosing people by early next week. But just to be safe, if you want to have an appointment, set something up for July 20th or July whatever, sometime in the July 20s at your convenience, and chances are we'll be up and running. Yes, sir. We're the same company, yes, and we're all cross-trained, yes. Yep. Different actual nurses, but we all work together. Yeah, with the staff, I'll be available. Obviously, I might not be able to meet all 500 people personally, but I'm certainly available for questions. And one of the reasons we we're having these educational sessions, we'll continue to have them, and so people can talk to me and ask their questions and make sure they understand things. And we want to be very clear about what to expect and what not to expect. So again, I'm the doctor in charge of the program. So at the end of the day, any issues, let me know. But hopefully you'll do well. We have some extraordinarily well-trained people here. So those are the people you'll be working with on a day-to-day basis. Well, Robert's one of our nurses in the back, and he can get you an appointment time right now, if you like, or if we don't have your medical information, you want to go over it in detail, you probably have to wait a little bit today, but Robert, I'm sure, can take that information or give you a form and fill it out, and then we'll look it over. But basically, we just want to make sure that you're the type of person that will be able to elicit an immune response and not have any serious illnesses that would put you at undue harm. Again, if you have some medical problems, that's not an issue. If you have COPD or you had a heart attack years ago, that's actually good for the study because they're more vulnerable people that we want to treat. On the other hand, if you have something like leukemia or something that could affect your blood cells, then you're probably not a good candidate. So those are the kind of things we look for. Make sure that we're not putting anybody in harm's way. Anything else? All right, Robert, I'm going to hand it off to you. Thank you, everybody.